And greetings, welcome to the Evidence Reasons Academy. I'm joined again by Katie, and we're going to do a little bit of journal clubbing on localization signals. She found some very interesting proteins. We are, unfortunately, we can't just decide, hey, we're going to find all the localization signal in this protein in this particular organism because um, it really depends on the amount of research that was done, and that costs a lot of money. So sometimes we just have to take what we're given. I'm very grateful she found information on the SOX2 protein and also P53. And so we're just going to do some journal clubbing. She was eager to share this paper with me, and because she has a far more beautiful voice than I do, and it's just wonderful to hear her speak, I'm going to let her share the papers she found, and we're going to read them together. So, Katie, take it away.
Okay. Which would you like me to start with? Do you want me to start with like the topoisomerase ones or the P53?
Why don't we do P53? A lot of the audience has probably had enough of topoisomerase for several years on my channel. We'll do P53.
Okay. So can you see my screen? I can't. Did you say something? Is everything good?
Oh, I'm sorry. I muted myself. Just wanted to make sure, is this fit for public presentation here?
Yeah. Can you see everything?
Okay, I'm going to bring it up. Hang on. And I'm going to put it on the main screen. And so far looks good.
Okay. Well, I have a bunch of articles that we can look at. I guess in our studies or our researching, I found that P53 has quite a bit of known nuclear localization signals. So here's just two like introductory articles that I found that kind of talk about P53, I guess.
And it's in one of your favorite model organisms.
Yes, I did. Yeah, this one was in Drosophila. Um, I guess I like these because they're nice highlights. But I believe it's used in apoptosis, correct?
You'll have to tell me. Every protein biologist I know tends to specialize in one or two proteins for their whole life.
Mhm.
I know that's kind of sad because each one is so complex. So, um, as far as these other proteins like P53, you can just kind of regard me as an amateur because I'm just a total newcomer to this. I've heard
Okay.
it like in a 1 hour lecture, a little bit of it, like only 15 minutes of the lecture. So I'm probably going to learn more about P53 through your reading of this than I ever knew.
Okay. Do you just want me to read through the abstract then?
Uh, we'll start out. Why don't you read through the highlights and then the
Okay.
abstract, and then we'll think about it a little more and
Okay.
reflect.
Um, all right. So, P53 tumor suppressor has become the most studied gene of all times. P53 is similarly regulated in Drosophila and mammals. P53 plays a conserved role in regulating metabolism and tissue homeostasis. P53 plays a non cell autonomous roles in both Drosophila and mammals. Non canonical functions of P53 have a potential role in tumor suppression along with normal animal physiology.
Whoa, whoa, whoa! Can you go back up?
Oh, yeah.
I did not know this. P53 tumor suppressor has become the most studied gene of all time. I did not know that. I thought topoisomerase was the one. Oh well, I've learned something.
I mean, maybe it's biased. I don't know how you would know something like that, really, but okay.
Okay, let's go through the abstract. That was great summary there.
Okay. So P53 is a central regulator of cellular responses to stress and one of the most frequently mutated genes in human cancers. P53 is activated by a myriad of stress signals and drives specific cellular responses depending on stress nature, cell type, and cellular context. Additionally, to its classical functions of regulating cell cycle arrest, apoptosis, and I don't know if I
Senescence.
senescence. Okay. Newly described non-canonical functions of P53 are increasingly coming under the spotlight as important functions on not only for its role as a tumor suppressor, but also as its non-cancer associated activities. Drosophila melanogaster is a valuable model to study multiple aspects of normal animal physiology, stress, response, and disease. In this review, we discuss the contribution of Drosophila studies to the current knowledge of P53 and highlight recent evidences pointing to P53 novel roles in promoting tissue homeostasis and metabolic adaptation. Okay. I guess I can do. I'm not sure where in the paper I saw that it had stuff to do with nuclear localization, but I could do like a quick Control F. Okay. Huh. Oh, wow. So it has some information on nuclear localization. I think somewhere throughout the paper it said that it has a couple sequences. Yeah.
Okay,
Or,
Katie,
yeah.
I'm sorry to interrupt you. My computer froze briefly.
Oh, okay.
If you have your telephone near you,
Yeah.
I can text you in case my system freezes so bad I can't even turn on my microphone. That's what happened for about a whole minute. I began to panic.
Oh.
So
Did you?
where were we? What were you? Um, so if I suddenly seem unresponsive, it's probably because my system locked up.
Oh, okay.
Um, we are not live. We're recorded right now, so this is not a catastrophe. If it does
Okay.
happen, I just don't want you to. I just don't want you to totally panic and think I'm ignoring you. It could be a technical breakdown. Um,
Okay.
will you be able to hear if I buzz your text, your
Yeah.
phone?
I
Okay.
have my phone near me.
Okay. So can we? Can you restate what you just said about like the last minute? I am so sorry.
I mean, I was just trying to find where in the paper it was talking about nuclear localization signals. They have a diagram here. I want to say that I read that P53 has, I guess it has 3 isoforms. Are these the isoforms? Or is it 3 separate
It's with the
nuclear?
P53 isoforms and protein domains.
Yeah,
Okay, so can we? I'd like to have a teachable moment here.
yeah.
Um, because we talked about it's very studied in humans and all sorts of creatures. So just to repeat, um, you know, we are very similar to chimpanzees in
Mm-hmm.
structure, and that can disturb a lot of people who have who are creationists because it's like, well, that looks like we're evolved because we have so much similarity to other creatures. But one thing this is a good example of why it's good that we're similar to other creatures. Can you imagine if we had to dissect human beings to find out how their parts worked rather
Mm-hmm.
than fruit flies? That'd be
Yeah.
bad. Okay, so just look at this as a gift that we call these things model organisms. We are presuming they model enough of how human biology works that we could still learn a lot from it
Mm-hmm.
without having to break apart the human. And in many cases, it's good we have simpler organisms we can break apart because then, um, if you want to focus on one thing, like say transcription or translation, it's much easier to study bacteria and yeast than it is to study the complexity of like a human cell, because there's so many cell types in humans. I mean, it just boggles the mind. So let's get back to the diagram. Go ahead and reshare it. I just wanted to make sure. I want to, there are a lot of creationists who study biology and they see the similarity of our similarity across all creatures. And I say, look, that's either common descent or common design or both.
Mm-hmm.
If one believes the evidence given for a young life and young birth, then it's definitely common design. And I'll just leave it at that. But I just felt it important that I mention it. So go ahead and let's get back to business here. Sorry for the total aside.
All good. So
Go ahead and I'll reshare it. Hang on.
oh, is it not sharing right now?
Let me see. Okay, there it is. All right, that was on my part.
Oh, okay.
I didn't realize I'm the one that had to do a click.
Okay.
So why don't you explain for the audience what an isoform is?
I can I'm not fully. I don't have like an actual.
Okay. Why don't I help you? You'll eventually see this, especially if you study, I think, molecular biology. And again, molecular biology is a bit of a misnomer because the real molecular biology is biochemistry.
Mm-hmm.
Molecular biology deals with transcription and translation. So in prokaryotes, you kind of read, with some exceptions, you read off the gene and transcribe it all into one RNA, and the RNA doesn't need a lot of processing, and then it can go to the ribosome and be translated to a protein. In eukaryotes, especially like humans, the genes are gigantic. So maybe, just as a rough estimate, and this will vary depending on the gene, 10% of the human gene, you extract only about 10% of that RNA, and again, the figure varies, and that RNA gets translated into a protein. So what happens to the other 90%, or even more? I've seen figures that are even bigger than 90%. Those are introns. Have you heard the word exon and intron?
Yeah.
Okay, so you can take a gene will have multiple exons, and sometimes you can stitch together kind of the same basic protein, but you can change a little bit of its composition by selecting which introns to stitch into the RNA, into the messenger RNA. So and so those break up into isoforms.
Mm-hmm.
We don't completely understand what all this does. It seems to be very important, but we're only beginning to start to catalog this. To understand how many, and so we call that alternative splicing.
Mm-hmm.
So we splice the gene up in different ways, and you get kind of a variation on the gene. The current estimate, as far as estimates go, and it could be way off, is that there are 20,000 basic genes in the human genome, but 500,000 alternative splices or isoforms. It's gigantic.
Yeah.
So I think this is showing the three major isoforms.
Okay.
So go on ahead, and we'll try to decode what this says. Oh, by the way, this is really good. See on the left, you have the amino terminal. That's usually what, if you're reading it in, like when you had those FASTA sequences.
Mm-hmm.
The start of the protein is the amino terminus. The end of the protein, if you're reading it kind of from start to finish, is the carboxy terminus.
Mm-hmm.
So if you wanted to say amino, that's kind of the start, and the end is the carboxy. So that was probably more for the readers. So go ahead and just go on here. So those are the It looks like the various isoforms and domains
Yeah, and it gives the number here too,
with the coordinates.
the
Very
coordinates.
good.
So,
Oh, can you scroll back up
oh yeah.
just a little bit? Okay, so I'm counting. I could be miscounting this. So I have 1, 2, 3, 4, 5, 6. It looks like 7 isoforms.
Mm-hmm.
I'm guessing.
Okay. Yeah, it looks like it.
And so there are these things called protein domains. Um, they're kind of like distinct parts of a protein. I don't know that there's a hard and fast rule of what defines a domain.
Mm-hmm.
Um, Sometimes we're able to identify domains because they do correspond to an exon, not always. And then sometimes we'll find things that are similar between proteins that are, they're not the same protein, but they'll have similar regions, and we'll say that looks like a domain. That's
Mm-hmm.
sort of how we define it. I don't know that there's a hard and fast rule for it. I will say this one thing we do know is when we look at the 3D structures, we can actually see kind of something that does look like a, kind of a physical, almost like a physical part. So that's sometimes how they might define it. I don't know that there's been a hard, fast rule, but it seems to be something we intuitively know to call a domain. So there you have it. So we can move on. Do you have any questions or comments?
Not about this. I think it's pretty straightforward.
Oh, I got it. I got to show you something. I just saw it. Is
Okay.
there a way you can zoom a little bit in? Is there a zoom button? Oh, wow. Very good. You see that? It says DNA binding domain.
Yes.
So from that looks like coordinate. I'm just looking at HSP53, the very top one. That's because that's just the easiest.
Yeah.
So from about coordinate 101 to coordinate 292
Mm-hmm.
is the DNA binding domain. That's a lot of amino acids that are going to be in contact with DNA. So
Mm-hmm.
this is a protein that actually interacts with the DNA, which is kind of amazing because you need DNA to make this protein, and then the protein will go back and and have contact with the DNA. That's a lot of amino acids contacting the DNA. That's amazing.
Mm-hmm.
And by the way, congratulations, you found the coordinate for the nuclear localization signal at 292 in
Yep.
the ASP53, and in the DMP53A through E, it's at 383. That's amazing.
Mm-hmm.
Well done.
Thanks. Yeah.
That is okay. Maybe that's worth reporting on. I, wow, you blew my mind with that one. Well done. Okay, so let's look at post-translational regulation. Now, this has nothing to do with nuclear localization, but you can zoom in on this.
Yeah.
Oh
Can you
my,
see that pretty well?
yes.
Okay.
So HSP53 is one isoform, and that is amazing. Okay. Oh my goodness. This is an amazing table. So uh, I hope you'll forgive me when I ask you some questions. It's just to kind of jog your memory, and
Mm-hmm.
it's kind of my way of helping to teach you, or kind of just review. I hope you don't mind me being that way.
It's okay.
All right, you'll tolerate it. So, can you describe? Okay, can you talk about post-translational regulation or
Yeah.
whatever you know about post-translational?
So after it's been translated, it's I have like a picture in my mind. So it has the histones, and like the DNA is tightly coiled around the histones, right?
Okay, so
Or
I'll help you out a little bit.
okay.
Histones our proteins, and they're subject to post-translational modification.
Yeah.
That's actually one of the most studied examples. But we're finding out in humans, we're loaded with post-translational modifications for
Mm-hmm.
all of our proteins, most of them, many of them. How about many of them? Okay, I
Okay.
did not know that the fruit fly, that little tiny fruit fly, would have all these post-translational modifications. So
Mm-hmm.
it's color coded there.
Yeah.
So we have, let's see. So the way they do this is when they say S6, you can see that S6, uh, I think S stands for the kind of amino acid,
Mm-hmm.
and I think that's a phosphorylation. Usually a
Yeah.
serine is phosphorylated. So T18 is a threonine. That's phosphor. Yeah. In eukaryotes, it's serine, threonine, and uh, tyrosine. I don't quite know. They get phosphorylated. Definitely serine and threonine. So that's how it's coded there. So tell me, can you see K120?
Yes.
Tell me what you think that means.
I don't remember what the protein is called, but it's getting acetylated.
K is for lysine. Very good.
Okay.
I mean, after a while, this is going to start to feel just natural.
Yeah.
Um, let's see. How about? Oh my! Look at this one. The K370. Oh, this is funny. Look at all the chemical modifications on that poor little amino acid.
Yeah.
So K is for lysine.
Lysine.
And why don't you tell me what post-translational modifications you see?
Okay, so it looks like it's getting acetylated, ubiquinated. I don't know this one, netylated,
I've
and
never heard of that. Okay, I've never heard of that one. That's a new one.
okay, and
And,
methylated.
and just when you think you know it all, you get humbled.
Yeah, I've never heard of that before.
um, those are so. That's amazing. They could stack them on that way.
Mm-hmm.
It could be that maybe they're not stacked, but you could find one or the others. And so this is what's interesting. Look at all the machines there on the right that accomplish this.
Mm-hmm.
So to get a phosphorylation, it can be achieved by any or all of these enzymes: ATM, ATR, DNA, PK, CK1, CK2, CK, CHK1 slash CHK2. Now, uh, again, can you imagine how difficult? What would these enzymes have to do to phosphorylate? They'd have to find the protein, right?
Yeah.
They'd have to go to the right amino acid because they're not gonna phosphorylate every serine because
Mm-hmm.
you can look at it at DMP53. You see, either they've not detected it or it just doesn't modify very much.
Mm-hmm.
So it has to decide: am I, should it be phosphorylated, and it has to locate it. Can you imagine the addressing system needed to do that? It's amazing.
Mm-hmm.
I know I'm repeating myself, but I continue to be amazed. So that wasn't about so much about nuclear localization signals, except did you notice between 292 and 331, there are a
Yeah.
lot of those translational modifications. Okay,
Mm-hmm.
if I had to guess, this might regulate possibly how how much of it gets into the nucleus. Meaning it could slow it down, amplify it. I don't know,
Mm-hmm.
because it's right there in the nuclear localization signal site. That's the first time I saw that. Can we put this as a note? Yeah,
Yeah.
some sort of note. Um, I don't know how you keep notes. Um,
I
The
can just
nuclear
write it down.
localization, yeah, the nuclear localization has post-translational modifications.
Okay.
That's an interesting find. First time I ever saw that. We may not know the effect of that. By the way, can you imagine how difficult it is to detect a post-translational modification? You'd
I'm
have to,
sure it's pretty hard.
yeah. So they'd have to extract the protein.
Mm-hmm.
So you take, so the first step is to take the cell out of the poor little fruit fly,
Yeah.
and then extract a P53. I guess you'd need a good centrifuge. Have you worked with centrifuges?
Yeah.
Oh, I'm jealous. So then you're able to extract and make sure you do have the P53 and that specifically the HSP53 isoform. So I don't know how long that's going to take to do. And then I don't know how they're going to detect whether it has a post-translational modification at site number 320.
Yeah.
Uh, I don't know what sort of chemical technique they have to use. Um, I got the impression from someone that studied it on histones
Mm-hmm.
that it was a miserable process.
Yeah. They actually study a little bit of this in the lab that I am in at school, but I'm not on that team. I'm on a different team. But I know that they were studying like methylation, and specifically with the set 8 protein or the enzyme, I
Wow.
guess.
So
Yeah. But I
actually,
think
I'm sorry I interrupted you.
I'm not sure if they were able to like get all of this information out, but I know that they definitely did a phenotypical analysis of the flies that had this. I want to say that the enzyme was completely taken out, maybe, or what happens to the fly when it's taken out. It was like a phenotypical analysis.
Well,
Um,
that's sometimes how we find out the hard way
yeah.
why it's important.
Mm-hmm.
Um, I will tell you because there's what we call redundancy. So, like the best illustration I have about redundancy is like the space shuttle. It,
Mm-hmm.
its navigation system is important for it to be able to fly because, like, especially when they're going into reentry, you have to be very precise. So they
Mm-hmm.
have 5 navigation systems. If one gets knocked out, the other 4, any of the other 4 could take over.
Yeah.
That's how deeply redundant it is because it's so critical.
Mm-hmm.
Similarly, in biology, we don't always, I'm just putting this out there, we can knock stuff out and the system will still continue on. That's a good feature. So
Mm-hmm.
sometimes it's not always, until you knock something else out, you may not actually know how valuable something else, one
Yeah.
thing is. Unfortunately, for eukaryotic organisms that are complex, I've heard figures of being redundant by about 7 levels deep. And you have to knock out an awful lot of stuff to be able to maybe guess. I'm just saying. But
Mm-hmm.
well done on your in your school that they're researching this.
Mm-hmm.
Uh, I would be curious to find out how long it takes, how difficult the process is to determine a post-translational modification at a particular. I mean, this is like getting a needle in a haystack.
Yeah.
Bravo, bravo. Anyway, um, so we have these post-translational modifications in the nuclear localization signal. Was there anything in the paper else that you really thought was interesting?
Um, I haven't fully like really looked at the paper. I just kind of saw that it had some stuff to do with nuclear localization, so it
Okay,
might have some more, but I'm not sure.
well, I tell you what. Why don't we just stop here? Because
Okay.
we only have limited resources, and even though this could be a fun paper to read from top to bottom, we might just skip it in the interest of time. So
Yeah.
let's look at just scroll down a little bit and see there are some symbols here that maybe it's just as well. Um, we don't go through because I'm not familiar with them and I don't want to mislead you.
Mm-hmm.
Uh, See, they have this thing called Delta 4 P53 and Delta 133 P53. Like if you read like a few lines down, I would feel uncomfortable trying to even comment on what that means.
Yeah.
Um, there are that may be variants, but everyone has their kind of their notation, and you need a specialist like people in your lab to be able to read that
Mm-hmm.
for you. Oh, okay. And I just scrolled down and it says nuclear localization signal NLS in orange.
Mm-hmm.
Okay. Um,
I think that's for B. I think it's talking about this.
do you want to try to extract a new? I would like to try to get maybe one sequence. I didn't know it was that precise. The problem is, you want to try?
Yeah, we can try.
We can always try. Hopefully, we'll be more successful than last week.
Yeah.
Okay, so um, why don't you? Let me see. I'll take this. Hang on one second. I'm going to try to bring my screen up.
Okay. Do you want me to
And
stop sharing?
yes, stop sharing, and I'll try to
Okay.
bring mine up. By the way, thank you for reading that.
Yeah.
And I'm gonna try to bring this up. I'm just gonna guess. You still have the paper handy, right?
Yep.
Okay, and you can guide me through this.
Mm-hmm.
So let's go to P53 drosa. How do you spell it? Like that?
Yeah, that's right.
Now the problem is we have 7 isoforms to work through,
Yeah.
and oh my goodness, and they have variants of the isoforms, and they haven't labeled the isoforms. Remember the labeling that they gave? HSP53.
Yeah. Mm-hmm.
Um, so you have the gene name, which will go to the corresponds to the DNA, but they may not necessarily have standardized isoform.
Hmm.
Oh boy. Okay, we'll just go. Okay, I'm going to avoid the thing that they say variant because I think so. You have a consensus sequence, which is
Yeah.
like most of them, and I think the variants are kind of the less represented. Okay,
Mm-hmm.
that's just a total guess on my part. This is why you're lucky that you work in the lab. You have context. They could tell you, hey, you totally grabbed the wrong one. So, um, all right, you catch me if something doesn't make. Um, you can expand your screen so you could actually just it'll fill out without all the, it'll fill out with what with just the Uniprot website. You won't have all the other stuff that's StreamYard related.
Yeah.
Okay. And I'm sorry, I'm just going to stumble like this. If you see something of how I might search better, you let me know.
Okay. The only thing that I'm worried about is that it does say isoform E, and I don't know, like, because in the article, it gave us two different coordinates to get to it. So I'm not
Exactly.
sure which one. I mean, if it's isoform E, it's probably on the second coordinates, but I don't know if it would coordinate with each other, I guess.
Okay, one way we can guess is we have to look for basic amino acids.
Mm-hmm.
That was a long nuclear localization signal, by the way, from
Mm-hmm.
coordinate, what was it, 292 to 331.
Yeah.
So that would probably be a bipartite where you have, do you remember the basic amino acids? Lysine, arginine, and
Histidine?
very good. And you'll have on kind of what I say the start of the
Mm-hmm.
localization signal, a whole bunch of basic, and then at the end also the basic. So it's kind of two. That's why they call it bipartite.
Mm-hmm.
And honestly, it used to be. This is what was so nice in the old Uniprod. I could find that if they had the nuclear localization signal, they'd have it. Subcellular location. Let's look at family and domains. Let's see if we went up to 292. See, it used to be they see where it says domain
Yeah.
333. It used to be that they would, if they had it, they would list it. Sometimes they may be reluctant to insert it.
Mm-hmm.
So I'm going to look for that arginine KKR. KKR. I know. Can you tell me? Okay, so each of these isoforms have different length, right?
Yes.
What's the final coordinate? Let's just go with the first one.
The first one, the final is 393.
Okay, so this one, I'm just guessing here. How am I not? I'm not reading this correctly. There's
Hmm.
got to be a way I could click.
Can you slide the top part?
Ah, very good. So I'm only able to get up to 334.
And the other one was longer. It was 495. I mean, it could be the beta or gamma version because those look a bit shorter, but it doesn't give a final coordinate for that. But yeah.
Can you imagine if we didn't have contact with the original researchers? We'd go nuts. So normally what would happen is if we really get stuck, this is why it's good that if you're working in the lab
Mm-hmm.
in a university, many times if you just write a quick note and say, hey, I'm working on this project. Okay, one, many of your um, like the professors there and the senior investigators, they would know other names. So if you can't get it through them, through your own, superiors, a lot of times they would be respected enough in the community that they could write someone a quick note and say, "Hey, can you clarify which entry on Uniprot this is?"
Mm-hmm.
Or can you give me the FASTA sequence?
Yeah.
It is, they'll be able to give it to you in just like 3 or 4 minutes. It just is a professional courtesy. Um, so so the nice thing is we have all these identifiers. Now this is funny why I'm seeing whether you get other names like you see the names that I have here. I don't think those are
Yeah.
the search engine probably found something more than we were looking for.
Mm-hmm.
P. What was the isoform name? HSP 53.
Let me look.
First one.
Oops. It was HS. HSP53. Yeah, HSP53. That's all.
Let's see if we can. No results found. Oh,
Do you want to try the other one, the DF
How long? How long was it? You said how many amino acids did you find?
393?
And I don't see any that have 393.
Huh.
Oh man, bummer. I was getting so excited. Is there? Can we go back to your paper? And so I'm going to take down. I'm going to stop sharing my screen.
Mm-hmm.
And if you could bring your paper up, let's see if we can get a little lucky. Otherwise, we'll just have to move on.
Okay.
I really thought it'd be a lot easier. So what can sometimes happen is they may not have uploaded it to this database.
Yeah. I'd have
Okay.
to look in the rest of the paper. Can you see it?
Yes. So let's take a look.
Okay.
See if do they have any uh, supplemental. If you go to the left, um,
Mm,
if you scroll down, see if they have.
they have concluding remarks. That competing interest.
No, they don't have it. Oh man, this is a good paper. You would think.
Yeah, I was trying to see if they had like the full number. Well, hmm.
And they had the coordinates.
Yeah.
Um, well, let me just take a shot in the dark here on. You won't see what I'm doing here. That's okay. Okay. No, I'm not getting. I'm having a hard time finding it. So, um, I tell you what, you're probably if you ever get in touch back with that lab, they would be the best ones to help you navigate that. You have the right contacts, and I don't want to waste. I don't want to waste the time on something that could be easily fixed given your contacts.
Hmm.
So, man, we were so close. That's why I was like, man, wait.
It
We
might
just,
just need a little more digging.
yeah.
Maybe. Yeah.
I mean, we know it's out there. We know someone has found it, but they didn't publish the sequence. I'm
Yeah.
like, it's so close, and it's like you could give us the sequence. You could give us an identifier. Um, um, we could take a shot in the dark. So one way to take a shot in the dark. Um, okay, before we move on, before I take a shot in the dark, because we've already spent an hour on this. This was very good.
Hmm.
Um, what was the other paper that?
On P53 or on
I mean, what other paper did you find that you found interesting?
I cannot remember why I pulled this other P53 one up. It must have something in it. Let me look. Okay, so this is the other paper. This one's going back to topoisomerase 2, though. Do you want to talk about that one? Because this one gives, I think it gives coordinates to get to the rat topoisomerase nuclear localization signal.
Oh, okay. Why don't I? Okay. You can, if you could stop sharing, and
Yeah.
I see 1192 on your end. Um, when it said at 1192, the coordinate 1192, is
Yeah.
that a nuclear localization signal?
It Let me read that a little bit more. I don't think that is. It says first we exchange the CTDs of rat topo to amino acids 1000. I think that might be the C
C
terminal.
terminal domain.
But
It's called the,
isn't it in the C terminal domain, the nuclear localization signal?
yes.
So it might um it might be that, because at the end of the abstract it says these results indicated that the C terminal domain of rat topo 2A containing the 1192 to 1289 region is involved in the regulation of catalytic activity by associating with RNA, as well as the localization to the nucleoli in interface cells. So I think that might just be the C terminal domain.
That's what it sounds like to me.
Yeah.
Um,
The problem with this paper was that I wasn't able to read the full paper. I was only able to read the abstract, I think. Let me see. Um, oh no, I can get to the full paper. Or no, I have the figures. Oh, okay. So for this one, it just gives the C terminal domain. I thought that it did say the nuclear localization signal.
and it mentioned it, but it didn't give the coordinates. So, I mean,
Yeah.
um, it, it's really hard to get the exact coordinates down. They kind of like,
Yeah.
no, well, we got this domain off and it didn't localize. It's like, okay, that's like, yeah, okay.
Mm-hmm.
But to get the actual coordinates, that is pretty tough. So, uh, one. All right, so we're going to report how difficult it is to get the exact coordinates.
Yeah.
Um, I'm sorry, Katie, for all the frustration, but this is research for you. And
No, it's okay.
okay, if I may say, sometimes you think, goodness, why didn't you guys report this? So I would just say, when they're under the pressure of getting a publication out, there are just all sorts of things that would be nice to be that they will just edit out. So no, either the editor is saying shorten your paper, or you just don't have time to report it. So there's a lot of things you think that should have been done that aren't, and there are usually reasons for it, which is kind of sad, but that's just the way it goes with limited resources. So I was just looking here,
Mm-hmm.
and I'm gonna look one last time if they have any localization. Immune system, DNA, metabolic, lipid, cell cycle.
Establishment of localization. Would that be it? But I don't see anything. That's responses to.
It's red.
Oh,
Uh, ah, drew blanks again. So that's
okay.
2 weeks in a row. We drew. mostly blanks, but we were so close to getting that other one. So I wanted to say we could take a shot in the dark. So I'm going to
Okay.
go back to that rat beef. What was it? Drosophila.
Yeah.
Let me just try. I mean, you'll never know what search terms might. It's amazing. See, I got a slightly different search results when I just put in.
Yeah. Fruitfly.
Okay, so this is a lot longer. The other one was 390.
495 is the number of um the second isoform that we saw. So that
Can you?
might
What was the number?
495 like was the
Second
number of the
isoform.
DMP, all of them. I think they're all 495.
Okay, now we have a little bit of a clue. This is where the detective work comes in. Um, it's going to be funny. Do you remember the post-translational modifications? Um, I tell you what. Why don't I stop sharing? I want to see that one with the 495. I'll tell you where I'm
Yeah.
going if you could bring it up. Now we're acting like detectives.
Okay. Can you see it?
Yeah, I'll bring it up. Let me take a good look. Can you scroll up just a tad, just a tad? So
Oh, you want to see these?
there's one that goes up to 495.
Yeah, which unfortunately is the one without all of the um post-translational
Okay, so I wonder what DMP 53 means.
um
Because do you see how it's saying the
Drosophila
coordinate codes?
melanogaster.
Okay.
So I think it's comparing the Homo sapiens with the Drosophila. Oh, I wonder why that's why we couldn't find it because these are Homo sapiens. Oh.
Okay. All right. Then, hey, how does it feel to be, learning the hard way? See, this is the thing: is if when you're with the laboratory and people have been working with this, or if you're kind of like a lot of times they'll have a conference, you get to talk to the researchers and they'll explain their papers. You
Mhm.
build a relationship, a friendship. Many times you could just send them an email and say, "Oh, I know so and so. I'm happy to respond." They won't think it like a spam. That's one of the nice things in the community. The
Mhm.
other thing is, since we're all talking about the same topic, it's like they love to talk about this stuff. They're happy to email. This is how they socialize.
Mhm.
So, well, we might have found something equally valuable. So let me see. Let me get on Uniprot again and human P53. Now let me see if I get a hit.
I'll
Okay.
stop sharing so you can share.
I'll share my screen. Is that all right?
Yeah.
Okay. And look, how many amino acids is the first entry?
393. That's crazy.
Score! Oh, now I'm excited. All right, so now I'll scroll down. Oh, look at all this. This is the most studied protein of all time.
Yeah.
I got that sad news for topoisomerase researchers. I'm just kidding. So establishment of localization. Look at this. Oh my goodness, look at
Got
that.
some.
We actually got a
Wow.
hit. There it is. Protein import into the nucleus. And so there is a term. Well, they have some data on it. I don't know how to interpret all this.
Oh, I think were those all different
I have
species?
no I have no idea what I'm looking at. It just
Oh,
says if you click on here where it says establishment of localization, I'm
yeah.
presuming that that gives you like the you know some data on how they established it or what it is. But let's
Okay.
now look, see if we get lucky here. Look at all those post-translational modifications.
It's a lot.
I know. Do you see all the interactions with all the other proteins?
Yeah.
It's just jaw-dropping. I mean, oh, by the way, P53 interacts with a ton of molecules.
Hmm. Well, I was just thinking about the apoptosis cascade and how much occurs in that, so I could see how much um,
Yeah, it's the
P53 would
most
interact with other.
it's the most studied protein of all time. So now I might have a new favorite protein. So let's look. All right. So my guess is we're going to find, what do you call it? The basic amino acids.
Yeah.
So this is, I don't like the way they numbered this. 292 to 331.
Well, do you think that it's because would 292 be the end of the DNA binding domain or the beginning of the nuclear localization? Or would it be like 292 to 330?
I think it's two, well, the way they might have notated it, but I need to look at something here. So this is
Okay.
one, two, so 10 is the last amino acid, and so 70 is the A there. So that's 292, 290, 291, 292 begins with a lysine.
Hmm.
Now, honestly, I would have thought it would begin with 290 because arginine
Yeah.
and lysine, but it's saying it began here
Hmm.
and it ended at 331. So,
I'm thinking that it might end at 330 and that the numbers were the start of each new domain.
okay. So, what we can do is we'll do blast. We're just gonna, okay. This is kind of dangerous here, but we'll.
Mhm.
I'm gonna put. Let's align two or more sequences. So I'll put. This is the localization sequence in a human, supposedly. Man, that's a. I, okay, so what I would have expected to see was like KKR or something, and I'm not seeing it here. I mean, they could be off by one because um, nuclear localization would normally have like a RKK or something like that. So I don't know
Yeah.
if they're off by a coordinate here. But then if they're saying it ends here, I don't see the way that it ends. It just, I mean, even with three amino acids, just by chance, we're just looking at the wrong thing. But if they have actually, where they knock this out, like you saw in those experiments.
Hmm.
So let me get another uniprot, and this time we'll get the, what was it, P53 of the Drosophila.
Yeah.
So let me get,
So it would
I'm just going to get the
whatever
big,
one was 495, I guess. Yeah.
so I'll go down here and look for, Okay, so I could download the sequence, and oh, by the way, uh, you can you saw what I did. I downloaded the FASTA sequence,
Yeah.
and it has all this other stuff that is just comments
Mm-hmm.
because it has this little greater than sign. Uh,
Yeah.
Blast is just going to ignore that. It's very helpful that you can insert comments into the
Mm-hmm.
sequence just to help identify things. So I'm going to look for this sequence in a human and see if we find anything like that in a fruit fly. And we run blast. Um, Remember, this is a very quick blast because it's going to look just for a very no significant similarity found. Whoa!
Wow.
So the human P53 is not very similar. We found a sequence that's not similar. That's claimed to be localization in the fruit fly. I
Mm-hmm.
think this is, you know, uh, it's almost we're almost out of time. I don't want to wear you out here. Um, do you think you'll have any more contact with your laboratory when you go back?
Yeah. I'll be in my lab next year too. So I do
Do
independent
you,
research.
do you get college credit for it?
Yes, I get credit for it.
Fabulous. Okay. Um, it's like um, I feel kind of bad even asking you for a favor here, but you can tell my sincere interest here.
Yeah.
Um, I would like to know. We have to meet someone that is more familiar with P53. I could probably meet some people
Mm-hmm.
in P53 in humans. Actually, oh my goodness, I know professional cancer researchers. I know guys who know P53 like the back of their hand for humans. And I can ask them, can you help me confirm that this is the nuclear localization signal in a human? And I
Okay.
could work that end of it.
Mm-hmm.
If I'm able to get that sequence and we're not able to find it at all in Drosophila, I think that'd be very interesting in and of itself. So we
Mm-hmm.
could say tentatively we found some of the reports. Our best knowledge doesn't see any homology at all, which is amazing. I mean, we might have done the search wrong here, but um, it's worth looking into. Now, when we were doing this as Aspergillus, that looked kind of, it wasn't convincing. So um, this is a legitimate way of saying it. Hey, we tried our best. This was our method. It could be flawed. We would like feedback, but this is given this method, we were not able to find um, homology, sequence similarity in the nuclear localization signals of humans versus fruit flies. And we got very marginal similarity between uh, Aspergillus and humans. And just leave it at that and say, look, uh, this is why we need to apply for more grants to
Yeah.
solve the problem, right?
Mm-hmm.
But it is an interesting question. Um, and no one really, this is a question a lot of people don't ask, but this will be a setup for an experiment where we could probably take an import tin from a creature like a rat and insert it into a poor little fruit fly, and if the thing doesn't even you know germinate, well, it's like, oh well, we found out how much coordinated change is required uh, to implement nuclear localization with the importance.
Mm-hmm.
So if you kind of remember what I was saying, if you change the important, you're going to have to change all the proteins. It's going to be a very severe change for the organism. It's not going to be able to. It's going to have a very difficult time evolving from one organism to another.
Mm-hmm.
Um, so this was very fruitful. I mean, it was kind of a headache to get there.
Yeah.
So I guess next time we'll know the difference between Homo sapien and a fruit fly.
Yeah.
We'll know kind of the convention. So I'll see you backstage in a little bit. Do you have any closing? Do you have any closing thoughts?
Um, Maybe just that this is a very interesting finding today. Um, I was surprised that there isn't any kind of significant correlation between the fruit fly and the um, human, but that could also be because this is a different isoform. Like there was different isoforms in the fruit fly, so I'm not sure. But yeah, that was pretty interesting. So
Well, we kind of know what questions to ask.
yeah,
Maybe we'll just try to ping as many scientists in the field that uh, would have the requisite expertise. I, I was actually shocked when it said couldn't find any significant similarity. That was shocking because
yeah,
when we did that with aspergillus, it could at least find like say 3 or 4, the three
yeah.
or four little amino acid sequences. So that did surprise me. Well, how about you could say goodbye to our audience, and then I'll just end the recording.
Goodbye.